Now let's move on to part 4 of this lecture, which is going to be about heritability. So the question that heritability actually asks is, how much of the phenotypic variation is available for selection? As I said before, look at our lovely Clint Eastwood and his son Scott Eastwood. I'm so happy that heritability is actually something that exists. So there we have our phenotypic variation that consists of our genotypic variation and our environmental variation. Now, heritability can be calculated by looking at that proportion of the phenotype that is passed down from the parent to the offspring, and that is the genetics. So we have the broad sense heritability, and that is calculated by dividing the total genetic variation over the phenotypic variation. However, as I told you before, the genetic variation consists of 3 parts: an additive genetic variation, a dominance variation, and an interaction variation. The only part of the genetic variation that is passed down to the offspring and that can be seen in the offspring is the additive genetic variation. And therefore, we have this thing called narrow sense heritability, and that is calculated by taking the additive genetic variation and divided by the total phenotypic variation. So let's take a closer look at that. So how in words can we interpret the broad sense heritability? Broad sense heritability is just the total genetic variation inside the phenotypic variation. So all the variation we see in our phenotype that can be explained by genetics. When we look at the narrow sense heritability, that is the part of the phenotype that is explained by the additive genetic variation. And this is the part that we can use when we select for animals. So this is the heritability, the small h squared. This is the heritability that we use in practice when we breed for cows for extra milk, or if we breed for immunity, or if we breed for a specific kind of wheat flour or whatever. And one important thing is that the small h squared is always smaller than the capital H squared. Oh, where are you? There you are. Key message. Okay, this you'll need for your practical, but also for your exam. Okay, so be careful, young ones. The heritability is not the same as the degree of genetic determination of a character. What does this mean? This means that, for example, when we look inside a laboratory rat line, when we have rats who are all homozygous for every allele, for every locus, that would mean there's 0 genetic variation. But that does not mean that they don't have genes that control fur color or that control the length of their tail. That still exists. It's just that the heritability is 0 because there's no genetic variation. So another one is that traits that are evolutionary important have a very low heritability. And I'll show you in a minute why that is. Another one is that traits with a low heritability can still have a very high, rate of genetic control. So let's take a look here at this image. We have here a population of, I believe they're called lemurs. There are these like cat-like monkeys that live in Madagascar. Now we see that we have a population that is with a lot of variation in fur color. And then when we look at the central picture, when we go to the left, we're selecting for very pale fur colors. And what you'll then see is that by selecting for a very pale fur color, at some point when you do that for long enough, all the animals in your population will be fixed for specific alleles in all the loci that control fur color. And what you'll get then is a population with purely the same fur color. Of course, there will be a little bit of variation because of the environment, exactly. But the thing is that your genetic variation, when you reduce your allele frequency and increase another allele frequency, you're gonna lose genetic variation. This means that if you select for long enough, at some point the heritability of your trait will become 0. So here's an example with fitness traits. So fitness traits can be like fertility traits. Now let's look at the fertility traits in these 3 species: mice, chickens, and cattle. For mice, litter size, heritability of only 0.15. And in cows, it's the worst. That's the conception rate, a heritability of 0.03. That's very low. But why is this so low? It is because for centuries, not centuries, as long as we exist, we have been selecting for fertility. If you select for fertility, what do we know? We know that these allele frequencies of the low fertility, that give you low fertility, they will drop like crazy. And the other one, for high fertility, they will increase. And what happens to our genetic variation when we start to make that difference between these allele frequencies huge? It drops. So what happened here is that for traits that are related to fertility, you're going to have a low genetic variation, a low additive genetic variation, and therefore you have a small heritability. So here we have an example with birds, and this is a real life example. And I'm going to make 2 observations about this graph. So what we have on the x-axis, we have the parents, and it's about the beak depth. No clue what this is, but apparently something about how big the beak is. And then on the y-axis, we have the offspring's beak depth. I make 2 observations. The first observation that I make is that parents with a big beak have offspring with larger beaks. And I also see that in 1978, beak size was higher, bigger than in 76. What does this mean? First observation means that it's actually a heritable trait. And the second observation means that over the years, they have been selecting for bigger beaks because we see a selection response. Okay, so here, this is just a small illustration of what it would look like. What does heritability look like when we compare the phenotype of the parents to the phenotype of the offspring? When we look at the left graph, we have a heritability of about 0.5, and what you see is that parents that are way to the right with a high phenotypic value often get offspring who also have a high phenotypic value. If you have a heritability of 0, like you can see on the right graph, you'll see that that parents with a high phenotypic value have offspring of the entire range, and offspring with parents with a low phenotypic value have the same kind of offspring. So there is no relation between the phenotype of the parent and the phenotype of the offspring. So there's a heritability of 0. Okay, so this graph again shows something similar. On the x-axis we have the phenotype of the parent, and on the x-axis, and on the y-axis we have the phenotype of the offspring. Yeah. Double check that I said that right. Okay, so over here, we have the offspring of the selected parents. So there on the x-axis, selected parents, y-axis, offspring of selected parents. Now I'm gonna ask you, what is the selection response? If O with a star is the average phenotype of the offspring from the selected parents, and O with a bar on top is the average of all the offspring. And then for the parents, it's the same. The P with the star is the average phenotype of the selected parents, and the P with the bar on top is the average phenotype for all the parents. Then you can simply say that the slope that you see here, that line, is the heritability, and that you calculate by subtracting the O with the asterisk. By doing O asterisk minus O bar divided by P asterisk minus P bar. And what this represents is the response and the selection. And next slide, right here. So what we hear, if we summarize it, is the heritability, the narrow sense heritability, is equal to the response divided by the selection. What we can do is we can turn this one around a bit by taking S to the other side, and then you can get the response is equal to the heritability times the selection. How can we calculate that? The response is equal to the average of the offspring minus the average of the entire population. And the selection is equal to the average of the parents minus the average of the entire population. Key message, important, important. So this basically shows us how we can use heritability, and how if the heritability is very low, you'll see that in your offspring, you're not going to have a big response. But if the heritability is high, you're going to see a big response in your offspring. So how is it that we actually, in real life, estimate heritability? Well, one way of doing it is by doing twin studies. For example, in monozygotic twins, so that's identical twins, what you know is that identical twins, their genetics are exactly the same. So all the variation that we see, that you see here in this left graph, all this variation is due to the environment. In dizygotic twins, which is non-identical twins, they're different in their genetics, and they're different in their environment. So all this variation you see here are a sum of the environmental variance and the genetic variance. Now, of course, these studies are super interesting, but we also know there are not so many twins. I think it's like 1/200 births that is a twin. And it's super complicated. What are we going to do in practice? Like, make identical twins in the lab, insert them into cows, and see if they're doing better than the non-identical twin cows? It's not a very great idea. So instead, what we do is we use SNP data. A SNP is a single nucleotide polymorphism, and it basically means that an allele that we know from previous work, it's just located in one nucleotide. And it just means that one nucleotide is different in one case and the other. In that case, we use this model here. B is a bunch of effects. It can, for example, be sex, or it can be season, or something that has a fixed effect on the phenotype. E is the environment. Oh, and that is my meeting. Oh, that is starting in 15 minutes. Let's click that away. Dismiss. Okay. Our G here is the genotypic value. And the genotypic value can be, if you want to write it in a slightly nerdy way, it says it's a normal distribution with an average of 0, and then it has a genotypic variation. This is the select escape. And so that is the genetic relationship matrix. And this matrix, in this case, we build it up by using all the genotypes for the different SNP. And that creates huge data. Now, back in the day when there were no SNP data sets and SNP chips and everything, we used pedigrees. And for the pedigree, the model looks basically the same. The only difference is the part that explains the genetics. And that is, before it was a G, now it's an A. And this A has a distribution here, and then we have an A matrix. And this A matrix basically contains all the relationships between all the individuals based on the pedigree information that we have. Now, the agrobiologists have built one of these A matrices some weeks ago during one of the TU on Friday. So this is where it is, and we actually still use it. It's a very useful tool. Okay, this is the end of this part, so let's move on to the next lecture.